This is warm welcome to all attendees and students and professors. This is Dr. Murugamani. Today I am going to brief us with that Hybridoma technology and monoclonal antibodies, which is the significance. Particularly, this monoclonal antibody significance in the sense it is much more efficient for the diagnostic drugs and the diagnostic drug diseases and treatment of diseases. Okay, from the basic onwards, that is immunosystem. Immunosystem in the sense here that is lot of microorganism in the environment. How we are combating with this microorganism based upon the term that is define and determine defense system is that is immunosystem. In this immunosystem, that is totally each and every human body is integrated. Particularly, that is immunosystem, all organs in the human body and cell to cell contact that will be integrated. How this combating with that microorganism? Microorganism in the sense there is a pathogenic and non-pathogenic microorganisms in the world. Here that is only we are focusing with the pathogenic organism. Pathogenic organism in the sense it is at three types. One is the bacterium, one another is fungi, one another is virus. In this one, how this different that is different particularly this is that how it is our body is protecting from the microorganism. First, environment. Environment it is in the human body, it is a totally it is attached with that human body in the surface of we can take it that skin. In the skin, how that is protecting. In the human body, that is a totally seven holes in the body. In the seven holes, how it is protected? This is primary, that is protection by the immunosystem. In skin, you can say that is a hardness and then salty, that is alkali. Effect it will be protect from that organism to invert into the human body. I, that is ITOs, which is containing lysozymes, which is killing the all organisms. In mouth, you can say that is a saliva, which is that having the saliva having the protecting that is enzyme, which is killing the organism and also alkali nature. When it is entering the food parts to the stomach, inside that is oxidic cells is secreting that acid, which is killing that microorganism. And each and every hole nose that is particularly the surface of the nose having that is mucus, that mucus is protecting that organism to invert inside the body. Then genital organs. Genital organ, even the urination also, that is a positive, that is urine positively, you have to increase the pressure, the urine has to come out. When it is the pressure is inside, that organism also, it will be protected. In genital organs, which is that is inside that also, that is some of that enzymes is secreting, that is immunoglobulins are secreting to protect and from the microorganism. Okay, this is one ear. Inside the ear, that is a wax, it is a produced, it is protecting. This and all that is a primary, that is protection by the immunosystem. Secondary. In this secondary in the sense, even though some of the pathogens that microorganism is entering into the human body, first it will be defense with that that is lymphocytes. Lymphocytes in the sense white blood cells. This lymphocytes having to how it is combating with that is fighting with the microorganism. This is totally integrated with that immunosystem that is in the blood, that white blood cells, particularly in the spleen cells, particularly that is lymphocytes. It is playing the main role that is with that all one how it is recognizing that organism, how this recognized organism is taken into the immunogenetic. In this immunogenetics, how this memory is within the B cells and T cells, and then how it is producing that is antibodies, particularly antibodies, how that is killing the microorganism inside the body. This is immunosystem. That is innate immune system in the sense from the mother to the child. We are getting that is immunity that is based upon the memory. It is already existed with the mother. It is reached to the child. That is innate immunity. Acquired immunity in the sense with the vaccination or previous infection of the particular microorganism that is acquired already immunity in the human body, that is acquired immunity. Okay. What are the systems in the immune system? It will be rolling the main role in the bone marrow, lymph nodes, lymphatic vessels, thymus, spleen, patches, and appendix, and adenoids and tonsils. This all the systems, not only this system, that whole body, each and every cells, it is playing the role to protect the pathogenic organism inward into the human body, particularly that immune system, that is how this protection it is originated from the stem cells. This stem cells is producing two type of cells. One is lymphoid stem cells, one another is myeloid stem cells. This lymphoid cells it is entering into the thymus organ. This thymus gland it is giving the T lymphocytes. At the same time, these lymphocyte stem cells directly it is producing naturally killing cells, NK cells, and B lymphocytes. This B lymphocytes is entering into the plasma cells. This B lymphocytes and T lymphocytes is playing the main role to protect the pathogens. But natural killing lymphocytes, it is playing the main role in the cancer cells. To combat with the cancer cells, how to that is subsidize the cancer cells, work everything, that biochemical work everything with that NK cells. This myeloid stem cells, it is erythrocytes and macrocytes and macrophages and granulocytes and monocytes. These all are the playing the role that is initially that pathogens entering into the human body, how it is accumulated and then digested in that particular pathogens. Pathogens not only that is aginated, that is we are calling that is antigens. Antigens in the sense that is a nature by protein molecules. This protein molecules is having the four lobes, alpha, beta, gamma. These four lobes it is based upon the antigen presenting in the particular surface of the pathogen. This pathogen, particularly where it is originated, how it is recognized by that is first recognized by macrophages, and then it will be engulfed by macrophages, and then some of the thing is monocytes, it will be converted into macrophages. The granulocytes inside that is a respiratory tract infection, that mast cells will be recognized in the particular pathogens, particularly that is a bacteria. That is recognized with the surface of the proteins, murine proteins, that is called murine proteins. In the it is in the surface of the bacteria. Some of the viral particles, particularly viral particles, that is a capsid, that is oligopeptides. It is in the surface. It is recognized by the moist cells, and then it will be producing the mucus inside the respiratory system. Like that, this all the cells, lymphocytes, natural killing cells, beta cells, and monocytes, and macrophages, and granulocytes, and most cells, and then all are is protecting the particular pathogens. Pathogens in the sense that is antigens is presenting cells, not only one, that is a foreign bodies. Any foreign bodies is producing, entering into the human body. It is the immunosystem is initiated. Okay, now this function, how it is initiated, how it is working with the tongue, that is first engulfed by the macrophages. It is becoming that when it is inside that engulfing that antigens, particularly that is a foreign body, that is recognized by the tongue, then it will be digested. When it is digested, the surface of the cells having that is APC, antigen presenting cells, it is having the surface. This is recognized by beta cells, this and also cytochrome T cells, that is T lymphocytes, which is having that is particularly stimulation of that B cells for that antibody production. This is memory cells. It is in reached to the plasma cells. It is producing that antibodies. Similarly, that is a cytotoxic T cells. It is producing that cytotoxic T cells. This is given to the memory cells. Memory cells, it is in the past more than the year, more than the month, it is rotation in that circulation in the blood, it will be protecting particularly that is the same organism. Again, it has should not enter into the human body. It is stimulation. The same that is the B cells that is defines against the extracellular pathogens to bind the antigen and making them easier to target and phagocytosis and with the complement. Again, this is that extracellular pathogens by the binding with the antigens, making them easier and the phagocytes with the complement with the T cells. When it is a communication with the B cells and T cells, that is the communication between the cells with the chemicals, that is intraleukins. Intraleukins also that is secreted by the T cells. It will be stimulate the B cells to produce the particular antibody. This antibody, it is genetically memorized in the gene, that is a chromosome 26. In the turn, that is more than 25,000 base pairs in the turn. This memory is lifelong memory and the short-term memory based upon the infection in the particular human body. Based upon the term, that is a HLA scale, it is given that is 6 types of, that is MIC, that is major histocompatibility complex. It is formed in the term that is based upon the antigen presenting in the antigen presenting cell surface, that is alpha protein, beta protein. Both it is the recognized by the particularly T cells and B cells, the surface of the antibody produced. This complement is how we are utilizing in the technology. That is particularly this treatment of the diseases is induced or engaged or suppressing the immune system. Particularly, that is already I given the topic, that is active immunotherapy. It stimulates the body own immune system to fight the organism. That is passive means that is it does not rely on the body, but it is attack of the diseases instead of immune system components that such as antibody or that is intraleukin, that chemicals is produced by the beta cells and create sometimes outside of the body, that will be giving that is immunity to the human body. Okay, Here, that is, we are explaining about the antibody, the outline structure. That is light chain and heavy chain. Both chain having that is a variables. This variables chain it is given by the long chain and short chain. In this long chain and short chain, that is based upon that is the base pairs presented one. Particularly that short chain that is 2000 50,000 base pairs molecule it is presented that in between the two short wings in between that is sulfide bond is presented. In the heavy side also that is long side that is a variable side constant side both region it is a both region that is attached with that sulfide bond. This is both short chain and long chain. That is the long light chain and heavy chain in between. That is a hinge protein molecule. It is presented. This hinge protein molecule is used to that is particularly recognizing the particular antigen. How it is antigen it is recognizing based upon the size variability that will be the hinge protein molecule it will be moving with the light chain. In the end of FA, that is light chain, that is FAB, that is antibody recognizing part. In this part, that is amino group is presenting. This amino group is recognizing the particularly active site of the antigen. The same time, that is FC in the constant variable, that is heavy chain. The end of the chain that is a single strand. The two strands in the end that is acid molecule. That is acidic molecule. It is presenting. It will be recognizing with that particular antigen presenting cells. This antibody is totally five categories. It is categorized by IgA, IgA, IgE, IgD, IgM, IgG. IgA alpha, IgA epsilon, IgD delta, IgM meta, IgG it is gamma. This five category of the tone based upon the heavy chain and light chain that is the molecule variability it will be recognized AG1 AG2 AG like that the variation will be conducted. This antibody derived from that is a beta lymphocytes, but this is classified into two groups. One is that is polyclonal, one another is monoclonal. Polyclonal that is mostly derived from the B cells. That is a batch to batch wise. It will be varied at the affecting that antibody reactivity and titer, but not powerful tool for the clinical diagnosis. Here, there is a monoclonal antibody is derived from single B cell clone that is offering reproducible and predictable and potentially inexhaustible supply of antibody with execute that specificity is much more. So it is easy to diagnose it in the clinical level. Enable the development of the sources of immunoassay system. Okay, now that is a production of monoclonal antibody by hybridoma technology. Hybridoma technology in the sense that a hybrid, which is that is the two variables, it will be cloned together, giving the new molecule or new variety of the cells. Here that is spleen cells, which is induced by the antigen, it will be producing antibody. That is spleen cells. For example, if you take it the mice, which is antigen, it is induced and then producing the spleen cells, which is 1 to 4 weeks. Totally, that antigen induced that particularly immunity system in the mice. Then that is myeloma cells. Myeloma cells in the sense that is a cancer cells which is that is cell lines. It is having that is H HGPRT, that is hypogenin guanine pyro, that is thyro, thiokinase, that particularly HAT media. It will grow, it will be diffuse with that particularly HK kinase enzyme. It is totally blocked with the tone, that is with that enzyme is called 8-axo 8-azoguanine. This 8-azoguanine, it is blocking that particularly, that is a hypothendin producing for that thymidine production. This is both are totally this spleen cells and myeloma cells. It is both are fused with the PEG media. It will be fused and then is cultured in the HAT media. HAT media in the sense hypothendin aminoputrin and thymidine media. In this media, only that is spleen cells cannot grow because in this cell, in this one having the thymidine. At the same time, that myeloma cells cannot grow without that is high GPRT enzyme, because this enzyme is playing the main role to produce the thymidine molecule. So it cannot produce. So that myeloma cells will be dying within the HAT media. Only that is a hybrid with that spleen cells with the PEG, it will be grow. That is cultivated and then again it will be propagated with that in vitro, that is a physical method, and then in vivo inside the mice itself it will be cultivated. Both are, it is again tested with that antigen, whether that is the activity of the particular disease will be manipulated with that harvest of monoclonal antibody. Antigen, when it is infected with that cell membrane and microorganism with the adjuvant, it is emulsified, it is injected in the spleen cell. With an titrated the serum, spleen is removed and then that is tested with that antibody production. This antibody is titled and then tested with that ELISA and flow cytometry. Both are, it is induced with that antigen, that is a booster dose, and then after 2 weeks, each and every day, that is spleen cells will be taken off and then for that analysis. This is screening of that mice, that antibody productivity within the mice. The cells will be prepared with that. The myeloma cells, it is treated with that 8-azoguanine or 6-azoguanine. This is treated with that one. Then it will be blocked with that HPRT enzyme, which is only will be viable with that high GPRT enzyme. It will be viable. Same, this enzyme is cloned with that PEG, that spleen cells, which is already, that is antibody protection. It will be taken place. It will be fused with that 8 of PEG, polyethylene glycol. Then it will be grow in the HAT media. In this media, only that hybrid cells will grow. These spleen cells and the myeloma cells cannot grow with that all. So it will be again, that is analyzed with that ELISA plates, and then that screening viable hybridoma cells will be produced. These plates will be checked with that all. The same on that hybridoma cells is a limiting dilution and they expanded with that in vitro, that is in bioreactor, that is we are cultivating the particular hybridoma cells for separation of the particular antibody. Otherwise, we are taking into that in vivo, same that is a mice. Inside that one, we have to produce and then we are taking out the spleen cells and then analysis with the ELISA or flow cytometry. In that hybridoma technology, all that is cloned, the selection positive hybrid cells are separated out and then cultivated in the positive clone. Here, that is how this is evaluated, how much it is useful. Particularly, that is this generation of that already in the market, that is a TNF antibody. TNF antibody in this human necrosis factor antibody. Previously, there is 100% mouse protein. It is produced. That is the first murine antibody. The next little bit, it is based upon the immunogenetics. It will be modulated. That 25% of the mice, that is protein, that is in the market. Again, it is only 10% of the mice protein. It is with that hybridoma technology, that is third. Now that is fully human based protein is produced with the hybridoma technology. It is in the market. That is Al-Dalal Moon. That product, it is in the market. Okay, what are the significance? That is a genetically engineered antibody is hepatocellular carcinoma. Particularly, that is it is defeated with that particular carcinoma within the antibody. It is a genetically engineered antibody in the market. Okay, what are the applications of the monoclonal antibody? Particularly, it is a diagnostic applications, biosensor and microarray, that is used for, that is a detection of particularly, that is what type of disease or what type of the disorders by the microorganism. Then therapeutic applications, particularly that is transplants, that is rejection, it will be used. And the cardiovascular diseases, it will be used in that particularly. Cancer also, it will be used to getting the result. Particularly infectious diseases, inflammatory diseases, this monoclonal antibody already in the market and then saving the humankind. At the same time, that is a clinical application, particularly purification of drugs, imaging the target of the drug, it will be used. Future application also, it will be developed. How much this monoclonal antibody in the market, it will be useful. This monoclonal antibody derived the development of multi-million dollar biotechnology industry. Particularly, this multi-million means this is the highest success rate, premium pricing, and potentially reduce the threat from the generics. Many of the leading pharmaceutical companies having entry in the monoclonal antibody sectors attracted by quicker and lesser costly development, high success rate and premium pricing, and potentially reducing the threat from the generics. The outlook of the monoclonal antibodies therapeutics is healthier, that outgoing success is existing products and combined with that bulging pipeline of new products awaiting for the approval and limited generic erosion and point towards the robust growth in the segment. Thanks for all participants and welcome for the next topic in the. Thank you.